All right, welcome everyone to our final webinar of Phenology Week. My name is Erin Postumus. I'm the Partnerships and Projects Manager here at the USA National Phenology Network here in Tucson. We're so glad that you're joining us today, and hopefully we've seen you in other webinars this week as well. We've been having a great time celebrating phenology and the science of the seasons with you all this week. We've had really great engagement on all of our webinars, and we've really been loving all of your social media posts and things and pictures and your stories that you've been sharing all week. So thank you so much for celebrating with us. If you're a Nature's Notebook observer, you probably have heard us say this many times that your data that you collect are part of a national database that are used by researchers. And that's really part of why we wanted to do this webinar today, to kind of showcase some of the ways that your data are being used and have been used by researchers. So we're really lucky to be joined by some researchers today who are going to be talking about that. Nature's Notebook data have actually been used in 166 scientific publications that we know about so far. There's a wonderful list that we have on our website. I'll put that link in the chat if you want to see the full list of all of the different ways that your data have been used. We also were really lucky that this year a colleague of ours at the University of Arizona here, Allison Meadow, did a dive into how those publications have been used in policy by policymakers and decision makers, and found that 20% of the publications have been used, which is way above the usual rate is only about 4% usually. So your data are very useful, and those publications of your data are very useful as well. And those kinds of reports are things like the National Climate Assessment, the Intergovernmental Panel on Climate Change reports, state wildlife action plans, species recovery plans, and a bunch of other state and national level documents. So it's really exciting that you have many different ways that you can look at how your data have been used, and we always try to keep you apprised of that through our newsletters and things like that. So I'll put the link to our newsletters in the chat too, so you can keep up to date on all the different ways that your data have been used. Today, we're very fortunate that we are joined by three of the researchers that have been looking at Nature's Notebook data. We have Dr. Rob Guralnick. He's the curator of biodiversity informatics with the Florida Museum of Natural History at the University of Florida. We have Dr. Ben Lee, who is the assistant professor of forest ecology in the Department of Biological Sciences at East Tennessee State University. And we have Dr. Janet Privé, who's a research ecologist with the U.S. Geological Survey at the Fort Collins Science Center. So thank you so much to all of you for being here. Just a few notes about the webinar before we get going. The chat is open, and we would love for you right now to put in the chat and let us know where you're calling in from. And if you'd like to share something about maybe a sign of the season that you're seeing right now, if you're seeing any signs of spring, or maybe it's still winter where you are, please go ahead and let us know about that in the chat. We would appreciate it if you have questions for our researchers to please put those in the Q&A. You'll find a Q&A box that's separate from the chat, and that'll just help us kind of keep track of those questions as they come in. We are recording this webinar, and we're going to be sending out the recording to everyone who registered. And then lastly, I did just want to launch a quick poll. I would love to find out from you all, are you actually a Nature's Notebook observer already? Maybe we have some local phenology leaders with us today, or maybe you're just a phenology friend, and I shouldn't say just, but you're a person who's interested in phenology and wants to learn more about the research. Maybe you're not a Nature's Notebook observer yet. So let us know. We've got responses coming in. We have quite a few Nature's Notebook observers with us, which is wonderful. We have a couple local phenology leaders as well. Those are people who lead local phenology programs across the country, engaging groups of observers to collect data for a particular purpose. And we have a lot of phenology friends as well. I'm going to end the poll and just share that with you all. So we have a good number of Nature's Notebook observers, so I'm sure that they're all very eager to learn about how their data have been used. So with that, I'm going to turn it over to our first speaker, Dr. Rob Guralnick.
Thank you, Erin, and I hope everyone can hear me okay. I'm going to share my screen. Can you all see the screen? Hopefully. I'll look for thumbs up from everybody on the chat. Looking good.
Yep, that's
okay,
good.
great. Okay. Hi, everyone. My name is Rob Gorelnik, and I am a curator and professor at the University of Florida and the Florida Museum of Natural History. And I'm really appreciative, honored, and privileged to be here with some amazing colleagues to be able to share with you some of the phenology work we've been doing over the past couple of years. So I want to just jump in and let's talk phenology. Um, well, first of all, phenology is such a great topic. I love talking about phenology because it is something we all experience. But I think one other aspect of phenology that resonates as well is that phenology is a sensitive indicator of change in the world. Um, in that sense, the fingerprint of change on the planet may show up most noticeably in phenology. And as I will discuss, changes in phonology can be like profound and surprising. So what we think of as spring now might not look much like what people considered spring 100 years ago, right? In terms of timing, in terms of intensity. But I think maybe I'm getting ahead of myself a little bit on kind of discussing all of these indicators and change dynamics. I think before I get to that, what I first need to talk about is how we can ever know about past phenology. Um, really, phenology data has been collected for a long time. I think maybe no example brings it home more than cherries and their use in monitoring over millennia. And the reason why we have such a long timeline for some cherry species is that cherry blossoming is really culturally significant. And in Japan, that blossoming is a signifier of the impermanence of beauty, or beauty of impermanence, um, because the blossoming only lasts a relatively short period of time. And so this sort of beautiful event that happens that lasts for the short period really resonates in the culture. Um, and to celebrate this cultural event, the blossoming of cherries, there have been festivals that have been popular for millennia. They've been held across different areas in Japan and the world. We have our own cherry blossom festivals here, and many of you I saw in chat were interested in cherries. And these are usually held at peak flowering. And these events were recorded by diarists and authors. And from these data, we can assemble amazing timelines of cherry blossoming. And here what we're looking at is 1,000 years of data, which like is mind blowing, right? Like we can see this huge record for the blossoming of cherries in Kyoto based on the timing of these festivals, ranging from 1812 to 2021. So we're talking like more than 1,200 years. Um, and what's really, really fascinating to me is that the earliest event for cherry blossoming, peak cherry blossoming, is actually the last record recorded. So it's 2021. There's an arrow next to a dot. That's the earliest recorded cherry blossoming event that was ever recorded. It happened just in the last year for this record, um, and you can see this sort of trend towards earlier cherry blossoming, earlier spring, if you will. And you kind of wonder what you'll see for these data in 2022 through 2025, right? As we think about those few years have happened since this record was assembled. Um, so that's like really exciting that we can look back across this record. But what about in the USA? What long-term data sets do we have here? We have some very famous site level data about phenology. And maybe the most famous of these is from Walden, where Henry David Thoreau went to live more simply. There's a beautiful quote from Thoreau on the slide. I really love reading Thoreau, and I've loved reading Walden. Thoreau was an accomplished naturalist who really noted the timing of plant and animal emergence very carefully in his reporting in his literature and in his diaries. And so from Thoreau, we have this record of timing in the 1850s, right? And these data have Thoreau's really I have a lot to say about Thoreau. And if you have questions, like he's a really interesting person. Um, but we these data really have served as a key set of information to understand how phenology in the Northeast has changed over time. And it really Thoreau wasn't the only one in the country who tracked phenology. And at many sites across the USA, people have recorded plant phenology, sometimes over decades, right? So we have these records, but they're often at just one place. It's much rarer to have consistent data over many, many sites over time and over species. And so while we have these great historical records, they're usually site-based, right? So that's been a challenge. But it turns out that some of these more multi-site level, multi-year, multi-species level data do exist. And it brings me to the key historical data I want to talk about today. And this data set was collected by Franklin Ho, shown here on the slide. Ho was a medical doctor, but also an avid naturalist. And later in life was actually appointed the first director of what would become the U.S. Forest Service. He is still known today as the father of U.S. forestry. But before he took on that role in the Forest Service and after the medical career as it was winding down, he pulled off something of a remarkable feat in my mind between that time period. So this is an interesting character. Um, he would end up running what might be the first U.S. community science project focused on phenology. And I think it's really exciting. So like what did he do exactly? Well, he built an army of volunteers to report what they saw in their towns and cities about phenology. And what he did is he set up a list of taxa, sites, and volunteers. And for those volunteers, he asked them to write down when they saw those key taxa that he delineated show first leaves or flowers or fruits. Um, his site and volunteer list is actually really quite impressive, and it stretched from Maine to Florida and out to the west to the frontier areas of our country in the 1850s. So this is a page from the compendium that he put together that shows the stations, the station names, the state, and the observers for each of those stations that were reporting on phenology. And then he compiled all those data um, all those observations into well-structured tables over all the sites and across those all the years that he was focused on this project from 1851 to 1859. So here's a scan shown on the slide for one taxon, Juglans niger, black walnut, and the flowering timing. And you can see those columns for the years. You can see the station names where these were observed, and that these data are all well assembled in this compendium in this document that you put together, right? Um, so it turns out like I had no idea this this resource existed, but Teresa Crimmins, who I think is sort of the Franklin Ho of our generation, in many ways, had come across a scan of this report and okay, it went down a little like this, right? Teresa was like, wow, this resource is amazing. And we can mine these data and ask some really cool questions about phenology change. Well, like I was blown away because the thing you really don't often see is this replicated sampling across sites, years, and species all in one spot, right? So I was like, heck yeah, we can look at trends spatially and temporally. So we both started thinking like, well, what do you actually do to make that happen? How can we begin to look at this historical record and compare it to the contemporary record, right? So we decided to take advantage of living in 2025. Um, Basically, we selected a set of species that had the most data in host compendium for leaves and flowers and we created clipped scans for those species and traits. Then we had to convert the data to a digital format, and for that, we ended up using an AI approach for converting these tables that we saw in that last couple of slides um, into digital text. Um, I, if you're interested, I can talk more about like the extraction and how we did this, but to kind of simplify, we basically saved those tables. We did a ton of validation because AI helps, but there's still a lot of mess that need cleaning up from these documents in terms of making it uh, as good as possible, right? And then we basically integrated those data and assembled them in a way that we could generate phenology estimates and phenology data in a statistical software package called R. Some of you might know what that is. So then, right, we have our integrated assembled data for the historical record. We need to think about how we're going to compare it to the present. And here's where we really leaned on the National Phenology Network and the amazing work that observers have been doing um, for many years to understand phenology and help compile the data about phenology. And we had about 13 species from the National Phenology Network that were well sampled that matched the list from what we had from the compendium from Franklin Ho. So we covered the rest using iNaturalist. There are five more species that we gathered through iNaturalist. We were then able to generate from those resources phenology estimations for onset of leaves and flowers from 2010 to 2023. Not every year is sampled for all species in the contemporary data, but that's true also for the historical data. I'm going to spare you the details of how we generated those estimates. It's important to think about because there are biases in these data, but suffice it to say, we were able to generate a pretty comparable set of estimates in the present to what Ho had generated in the past. Okay, so here's how that looks across space and across time. Oop. When we look at the estimates of phenology and how they're structured spatially and temporally, uh, we had to do a little extra effort to align the data over the same general spatial areas. But when all is said and done, we have a pretty rich data set across the eastern portion of the country, right? Um, so now we can actually look at these comparisons between historical and contemporary phenology across the region, right? And so what do we find? Well, here are some of these results. Here you're looking at the trends over latitude and day of year of timing of flowering and leaf out for our historical record and our contemporary record for flowering and leaf out over different biddings of whether the flowering and leaf out happens early in the season, middle in the season, or late in the season, right? But before we get too bogged down with this figure, I think what I'm going to do is zoom in into one panel for early flowering and leaf out, and use that to talk about what these results mean. Okay, so this is probably a little easier to dig into. The first thing to notice is that the yellow line is a lot lower than the blue line. A yellow line is a contemporary phenology trend over latitude in terms of the day of year of flowering and leaf out, and the blue line is the historical one. And this means that contemporary phenology is happening a lot earlier across all latitudes. And that difference is pretty large. It amounts to basically a month earlier than when Franklin Ho and his volunteers were looking at plants in the Northeast. And that's true for both flowering and leaf out, right? So like what Ho is seeing in his time period is actually really different temporally than what we see today in terms of contemporary phenology. One of the harder things to see in this figure, but it's there, is that the slopes of these lines are not quite the same for both the yellow line compared to the blue line or for the flowering compared to the leaf out. And that difference in slope of the yellow and blue line means that there's a bigger gap between historical and contemporary phenology timing in the north than in the south. And this is important because it means that phenology has shifted earlier, more strongly in the north compared to the south. And that difference is somewhere on the order of 5 days or more in the north versus the south. So the spring is occurring 5 days earlier relative in the north relative to the south, comparing contemporary to historical. Okay, right. So hopefully that's clear that there's a sort of trend for like accelerating phenology change in the north compared to the south. So you might be asking yourself like, well, why? Why is that? And we think the answer may be that land surface is warming faster in the north. Or really, that realized phenology difference is being driven by accelerating climate change in the northeast compared to the southeast. This is a figure from a paper from 2021 in Nature Climate Change. And what this is showing here on the left is that basically there's much stronger warming happening in the northeast, especially in the coastal regions of the northeast and into the sort of the Canadian boreal forests. And this warming has to do with a long-term trend driven by the North Atlantic Oscillation, which is shifting over the last few decades to bring more warm water towards the coast, which is warming the land masses and causing this trend, right? And this has led to more than a 2-degree faster warm up in the Northeast compared to the Southeast. And this is probably the reason why we see that phenology shift being faster in the Northeast, right? Okay, that's basically the story I want to tell you today. Thank you so much for listening. It's hard to see your faces, but I hope this was exciting and fun. Um, we covered a lot, but I have a couple more things I want to end with, just real quick here. Um, so I hope I convinced you that historical data and contemporary data like what NPN provides are incredibly valuable and can tell us about the rates of phenology change over space and time. I also only was able to share part of the story of the work we did in a recent paper in Communications, Earth, and Environment. QR code is on the slide. You can scan it and go right to the paper and learn more about what we did. I didn't have time to tell you all the cool things about that paper, but there's some really neat other parts I wanted to also give you a chance to look at if you're interested. And please take a look at that paper. I hope people get a chance to see how the whole story there. But mostly what I want to do here and ending is acknowledging the amazing work of my collaborators who made this possible. Dr. Lindsay Campbell, a close collaborator at the University of Florida, Dr. Teresa Crimmins, who needs to know more acknowledgement. She's just amazing at everything she does. Such a champion for phonology and been an enabler for so much great work. And an amazing graduate student, Erin Grady, who really was key for all of this happening and making it all work. So thanks to them. Thank you for listening to me, and I'm happy to take questions when we wrap up this session. Thank you very much.
Thanks so much, Rob. That was great. We did just have a kind of a clarifying question maybe we can get to before we move to the next speaker, and that's where are the data stored that were collected by Ho, and how did you come across those data?
Great question. Yeah. So the the whole of that document was produced, I think, for the Smithsonian or for the U.S. Congress. I think it was kind of like at that time period, it may have been the same deal. So that volume was basically a paper volume that was scanned. And then Teresa had come across the scan and was like, check it out. This is this amazing scan document. So we had to scan the document, then we had to OCR it, and then derive the data out of it, right?
So is that available for others to look at?
We are happy to share copies of that document for whoever would like to look at it. I also will say that all the data we extracted is actually available on as a digital data. So you don't have to go back to the original if you don't want to.
Thank you. All right, let's move over to Dr. Ben Lee.
Um, does that work for everyone? Can the presentation looking good?
Yep, looks great.
All right, great. Cool. Well, hi everyone. I am really excited to be here along such great friends and colleagues here, but also excited to talk to you today about phenology. This is something that I'm really interested in my research and really interested in just sort of getting the word out about how important it is as a signal and as a really strong driver of how ecosystems function and how plant populations in particular respond to climate change. So whereas Rob focused a lot on the history of the science and sort of the importance of historical records, I thought that I would too sort of focus on how the data actually gets used a little bit more. So what the important parts of the data are and why NPN data in particular is so important to the type of work that we do. A little bit about background about where I'm coming from and sort of what I'm affiliated with. I am currently an assistant professor in East Tennessee State University. That's about as far north and east as you can get in Tennessee, up in Johnson City. Prior to this, I've moved around a lot. I grew up in Seattle, moved to Ann Arbor, Michigan for grad school, did a postdoc in Pittsburgh at the Carnegie Museum of Natural History, and currently in Johnson City. I mention this all because one thing that I'm constantly sort of surprised by, or not surprised by, but just really interested in, and that's driven a lot of the work that I do, is just how different all of these places are in terms of the ecosystems and in terms specifically of the phenology. Right now, I have friends in Michigan that are still bundled up, haven't really seen anything. And down here, I just saw my first trillium emerging from dormancy yesterday when I was out in the woods on campus. So just really strong differences in where we see activity starting for the plants that I'm interested in. And specifically, I will also mention I focus on two main groups. The first is tree seedlings are my first love. I really like trees and trying to understand how forest dynamics are affected by phenology and by forest demography. But more recently, I've started branching out more into the spring ephemeral and wildflower communities. And so I'll be talking a little bit more about that today specifically. And as I mentioned, I'm originally from Seattle. I was lucky enough as an undergrad at University of Washington to grow up in forests that look like this. And as an undergrad, I was able to work up on Mount Rainier. And these forests are very near and dear to my heart. And what they look like is something like this. They're very dark, they're very wet, they're very quiet. All the moss really absorbs all the noise while you're out there. And they're very constant throughout the year. There is, of course, seasonality in temperature. There's seasonality in the amount of light availability. All of that's associated with local weather and the tilt of the earth and all of those great things. But for the most part, if you're a plant on the forest understory, the environment that you inhabit is relatively stable over the course of the year. It's going to be between about 40 and 80 degrees, probably closer to 40 and 70 degrees. It's going to be very dark. All of the evergreen trees above you are really going to shade you out. And because it's Seattle, because it's the rainy city in the Northwest, it's going to be very wet as throughout the year. Now, when I moved to Michigan for grad school, I was really sort of blown away by how different of a system the eastern temperate forests are from these Pacific Northwest forests that I really grew up in. And one of the main things that really stuck out to me was just the difference in the seasonality and the availability of resources, and just how different wildflower communities and plant communities in general look depending on what type of year you sample them in. And so one of the things that really drew my interest immediately when I moved up to Ann Arbor was my first spring there, just looking up in the woods and seeing how much light is coming through these canopies. And if you're in the eastern US, or if you're in another similar type of winter deciduous forest, this might be something that you see right outside your window or in the woods in your backyard. Right now, if you were to go out and look up, you're going to see a lot of sun coming through the forest canopy. Some of it gets intercepted by branches and by trunks, and maybe some remnant leaves from last fall. But for the most part, the amount of light is a lot higher reaching the forest floor than what it will be in a few months' time in the middle of summer. And this period, or this difference in light availability, is really substantial from the standpoint of an understory plant like a tree seedling or a wildflower. The amount of light available to an understory forest plant drops by about 1,000, up to about 1,000 times reduction in the amount of light. So, of course, all plants need light to fund photosynthesis and to generate the sugars that they can then use to metabolize and, grow, survive, and reproduce. And so in the middle of summer, it becomes a very stressful environment for anything growing in the understory. The canopy trees are fine. They have access to light throughout the year, but anything trying to grow below them becomes very shaded out. And so this sort of ephemerality, this temporariness of the light environment really drew my interest, and I became really fascinated by how the length, the duration of access to highlight availability affected understory forest communities. And of course, in a deciduous system, in a winter deciduous system, we have similar dynamics in spring and fall. But for me, I was really interested in spring phenology and spring access to light in particular. And spring is a really interesting time, both in terms of diversity, in terms of phenology, in terms of just the general ecology of these systems. If you go out into the woods over the next few weeks, depending on where you are in the country, you might see spring active summer green plants like the trillium pictured here. You might see erythronium, so trout lilies. You might see spring beauties. You might see dicentra, so Dutchman's breeches. I always forget the common name for that one. Um, You'll also start to see tree seedlings. So again, very near and dear to my heart is how trees recruit in the understory, and you'll start to see plants like maples and oaks come up from the understory as well. Besides plants, you'll also start to see the things that interact with these plants and interact with other parts of the environment. You'll start to see insects, especially pollinators like bees, both solitary bee species and the queens of eusocial bee species really rely heavily on nectar resources from spring ephemeral and spring active wildflowers. Uh, you'll start to see migratory bird species moving back into the forests. You'll see other insects and other animal interactors moving back and becoming active after a long winter. And you'll also start to see things like deer come back, which will use these plants as a source of food as they consume the wildflowers and the tree seedlings. And all of these pieces really come together into this nice, really cool mosaic or jigsaw puzzle, however you want to think about it, where they are all very adapted to be active at the same time and to mutually benefit, or at least in the case of deer herbivory, sort of get a one-sided benefit from that interaction. But you can imagine if any of these pieces fall out of the system, then the interactions that really structure the ecosystem as a whole also start to fall apart, and that can be really problematic. Now, one thing that we worry about as ecologists would be if any of these species sort of disappear over space, right? So we worry about extinctions or local extinctions. Does Trillium disappear from a specific location? But one of the things that we're starting to think about more is how phenology affects when these organisms are active, and if those interactions are ever affected by maybe Trillium comes out a lot earlier, senesces a lot earlier, and is not available for something that emerges a little bit later into summer. And so with all of these interactions really fitting neatly together under historical conditions, and then thinking about what Rob talked about in his previous talk of how phenology is changing, will those interactions really stick together, or will they start to fall apart and lead to ecosystem declines? Now, the phrase that we use to describe this is phenological mismatch, and I'll give you an example here of what I mean by that. So let's say that we have the phenology of a trillium flower here represented as this blue. Let me see if I can get a highlighter here. So represented by this blue distribution. So don't panic too much. This is not a math class. But you can think about this distribution as representing the duration of time that this trillium is active throughout the growing season. It will flower early in the growing season, and then it will maintain activity until late in the growing season. And we use this distribution to sort of represent not only a single flower, but also a population level. So the center of this distribution up top would really be emblematic of when a population of trillium would be at peak flower. Now, we can also overlay a similar distribution here in red, showing when bumblebees are active throughout the season as well. And given this historical perspective of how closely these two groups interact, what we would expect to see is that these two distributions are really tightly overlaid, that they are both active at around the same time, that they reach peak activity at around the same time. The flowers rely on the bees for pollination, of course, and then the bees rely on the flowers as nectar resources so they can gain the sugars necessary to fund their metabolism and to complete their life cycles as well. Now, the overlap between these two distributions, so basically from here where the red starts to overlap the blue, and here where the blue stops overlapping the red, the distance between these two lines represents the duration of overlap between these two species, or between the activity periods of these two species. And one of the things that I really think about in my research, and especially when I use community science data like NPN data for this research, one of the things that we focus on is whether this duration of overlap is changing in response to environmental change. And to sort of give you a couple of takeaways from this research, we know that things are moving earlier, like the cherry blossoms that Rob put up in the last talk. We know that things tend to be, at least for spring events, they tend to move earlier in the year, and that's generally very strongly associated with warming temperatures. What we know less about is if if different species are moving earlier at the same rate. So are they going to be moving together and maintaining these interactions, or is one going to be moving faster than the other, and will this interaction break down? And so, for example, if we know that wildflower phenology and bee phenology are both moving earlier, let's say that the wildflower phenology moves a lot more strongly in response to warming temperatures than the bees here. And what we would see instead is that the duration of overlap shortens. It's moved a little bit earlier in the early season, but it's moved a lot earlier towards the end. And so what we see is this shrinking in the duration of overlap, meaning that the overlap for the pollination resource from the flower's perspective or from the nectar resource from the bee's perspective, they're becoming less available in time. And so even if both of these organisms are still active in the same space, this phenological mismatch can lead to a breakdown in the timing and the duration of these interactions. Now, one of the things that I'm really interested in is plant-plant interactions. And so one of the questions that's really key to my research is what if climate change will alter phenological escape. And what phenological escape is that these wildflowers and other understory plants like tree seedlings will emerge from dormancy up to a few weeks, up to a month, month and a half before the canopy actually closes above them. And so if you remember back to some of my earlier slides, the amount of light that's coming through an open canopy is really valuable to these understory plants. And so rather than having historically a large amount of overlap between the activity periods for these species, what we see instead is under historical conditions, these wildflowers emerge really early. There's going to be some overlap with the canopy for a lot of these species. And then as both of these move earlier in the year, what I'm really interested in is will this duration of light availability, so which will start here with, sorry, that's not a super straight line, start here with when the wildflowers become active in spring and ends as the shade above them begins. This period of time of high light availability, will that change in the future based off of how these plants shift? So for example, let's say that this canopy is moving a lot earlier per degree of temperature relative to this wildflower. And so we start to see a lot more overlap in these two distributions. And so instead, the period of time for which these plants have access to high light is really shown here instead. We see a much shorter period of time, having trouble drawing a straight line with my mouse today. But we have a much shorter period of time where this highlight is available to these understory plants. But the opposite could also be true. Maybe the canopy trees are not as responsive, and so the period of time is actually moving to be greater from the understory plant's perspective. Now, I'm not going to talk too much about results or about specific projects, but I did want to drive home as part of my talk just the type of data that is necessary for us to be able to do these projects and really hopefully drive home why NPN is such a valuable resource for the type of research that I do. Some background about the research that I do with Phenology. I use a bunch of different resources, and I saw a question in the chat earlier about using remote sense data for GreenUp. That's something I use as well. One type of data that I use is observational data. I will go out and I'll go out with my students, and we will just monitor the activity of things as they come up in spring. My field season just started, so we are a little bit stressed and trying to get out there and make sure we capture everything. But that's definitely a source of data I use. A huge one is community science. And so especially NPN and Nature's Notebook, Rob mentioned briefly in his talk right before this, iNaturalist is another fantastic resource. This is an example of some of the observations I've made here in Johnson City over the past few weeks. Another thing I'll plug here is the Global Biodiversity Information Facility, GBIF, is a great repository for all of these types of data. So it contains both NPN data, iNaturalist data, herbarium data, which I'll talk about in a sec, and it sort of puts it all in one place and makes it very useful. when I was in Pittsburgh, I was working at the Carnegie Museum. I got really comfortable working with herbarium specimens. This is another great way to get historical information. So you can think of herbaria as sort of a plant library. So where plants were collected in the field, mounted to paper, and then were preserved with, you can see sort of down here in the bottom, they also contain information about where and when they were collected, and that allows us to be able to use them for this type of phenological study. The phenology itself is preserved in the specimen. You can see here this dicentra was in flower when it was collected. So if we know where and when it was collected too, we can match that up with environmental data and then use that information to model responses to environmental change. And lastly, as mentioned, I do use remote sensing in my work as well. One of the main data products I use is MODIS. I'm not going to worry about that for right now, but MODIS, MODIS. And one of the things that it tracks is green up over larger landscape level areas. And so that's a really valuable tool that I also use for some of my overstory tree canopy phenology as well. Now, plugging this into models and actually doing an analysis, we need the phenology observations, we need site location level data, we need date of when something was observed or collected. We need to know the species that was identified. This could go either way. If you're just interested in canopy chain or canopy closure timing, for example, that's not necessarily a species level thing, but for understory plants, it's very helpful to have species identity. And then we can take all of this information and plug it into data sets like PRISM and pull out environmental data associated for that time and location. And we can do cool studies. So this is just an example I pulled from the NPN website where you can look at, we can look at for American beech, for example, how the phenology differs year to year. So anything in green here was an observation of when beeches had leaves present. And you can see x-axis here is time throughout the year. And so we can look at differences between years in when these plants were active. And then we can track those differences between across time or across area as well. Now, I will also say within all of this data, one of the main limitations for these projects is observers and observations. I can go out with my students, we can collect data around East Tennessee, but if I want to do something more national or regional even, it becomes really challenging really quickly to be able to take these observations sort of simultaneously across a wide area. And so community science data sets in particular are incredibly valuable for getting us a lot of data very quickly in a very structured way, ideally. Now, organizations like iNaturalist, super valuable data, but because it's a lot more opportunistic, it's people going out and recording observations, but not necessarily in a very controlled way, and certainly not necessarily in a way that they're monitoring the same plants or same populations through time. We can use that data, but it becomes a little bit more challenging statistically to account for all of the noise. With NPN, it's super nice data, and it's really nice for us and really convenient for us to be able to use that data in our research. And just to give you a taste of what that can look like in terms of results, here, going back to this phenological escape question, one of the things that we can look at then is whether or not the period of high light availability seems to be changing in response to temperature. So here the dashed line is wildflower activity, and because it's a negative slope, you can see that the wildflower activity on average is moving earlier in time. And same thing for the trees. But because these lines intersect and the distance between these lines is decreasing, it's changing as we move from cooler to warmer temperatures, what we conclude based off of this wildflower example is that there will be a net reduction in access to light for these spring wildflowers, which is not good news if you're a wildflower. We can also even take this information and blow it up to international or global scale studies, like this study from 2 or 3 years ago, where we were looking at how phenological escape might change across Asia, Europe, and North America. And similar to what Rob noted earlier with how the Northeast will be particularly and has been particularly affected by climate change, we see that here too, where these dark blue colors represent areas where wildflowers are expected to experience the sharpest declines in access to light too. So not only is phenology changing the most rapidly, but it's also affecting some of these biotic interactions really strongly up there as well. And I just wanted to finish by thanking any of you who are our observers, or any of you who have been motivated this week. This Phenology Week has been so wonderful to be a part of. But thank you for collecting your data. We really use this data a lot. I don't have a clear number of how many observations that I've used from different observers across INAT, across NPN, but it's definitely over 10,000. So a lot of this work, it would just be impossible without the scale of contributions that you all make to this science. So thank you so much for your participation. And yeah, my work would just be impossible without you. The last slide I want to put up is just a plug. If you're interested at all in some of the topics that I talked about today, my research can be found, including free PDFs of all of the work that I've published so far on my website at peachlab.com. 2Cs was how I managed to get it to work out. And if you have any questions, please feel free to send me an email at leebr1@etsu.edu. And I'll take questions now if they're quick, or we can wait until afterwards in the panel.
Thanks so much, Ben. That was a great overview of phenological mismatch and a great plug for more data that are needed. So I think we're going to move to our last speaker, and then we'll see if we have time at the end for some more questions. There's a couple in the chat if you want to try to answer them directly. But let's move over to our last speaker, Dr. Janet Perfet.
Yeah, thanks. Man, those were such good talks. It's going to be hard to follow up, but I'm going to try and do it quickly so that we get done on time. Can everyone see my slides?
Yeah.
Okay, great. So I'm going to focus right now on recent trends in phenology observed across the United States, just looking at data collected through the US National Phenology Network. So a lot of the data that you all have been collecting. And I don't have to spend much time on these slides because the other speakers did such a good job talking about it, but we know that phenology is important, right? It's fascinating. All of the phenological things that we see are so visible. And like Rob said, these changes in phenology are among the most visible and sensitive indicators of climate change that we have to date. And shifts in phenology can have really widespread and cascading impacts on ecosystem processes. So phenology is important, right? And it's also important to collect good phenological data, as was also covered in the past two talks, so I won't spend too much time on this either. We know that historical long-term records of phenological data are rare, other than the cherry data that Rob Gralnick talked about from Kyoto, Japan. Here's another visualization of this extremely long time series of cherry blossom data. Herbarium images, personal diaries from people like Thoreau provide some of this historical phenological data that is very important. And modern records are more numerous. So as someone mentioned, satellite imagery, we can collect phenology data at long-term monitoring sites using time-lapse cameras, and also where you guys come in, volunteer scientist observations. And we wanted to focus on how those volunteer science observations, if we can see any trends in these observations that you guys have collected. The National Phenology Network was established in 2009. So in some cases, we have up to 15 years of phenology observations taken on the same individual organism or animal species at the same sites over long periods of time. And these observations that you all collect, they differ from other crowdsourced publicly available observations from, say, herbarium images or iNaturalist observations, because the frequency of these observations is also recorded. So you all note when you do not see a phenophase, and then you note when you do see a phenophase. So you have sort of this measure of temporal precision that researchers can incorporate into statistical models of phenological change. And that is what we're currently working on right now. So the results I'm going to show you are very preliminary, but over the past year or so, the National Phenology Network team and I have been asking whether we can see any overall trends in National Phenology Network data over these past years. And to do this, we decided to examine trends in phenology and climate over time, just for observation series contributed to the National Phenology Network that spanned 9 or more years, to get sort of the most robust signal over the longest time period within this data set. And here I'm going to talk about these observation series, and they're just series of observations taken on the same phenophase of the same individual plant or animal species at the same site over time. And we are only analyzing trends in the first yes observation. So the first time you guys see an observation of a phenophase per individual phenophase site in year. And we're also looking at how excluding observations without a prior no phenophase observation within the last 14 or 7 days influences the overall trend in results. And we're only focusing on phenophases that indicate sort of a transition from one biologically relevant stage to another for plants, especially. So like leaf bud burst or open flowers is what we're focusing on here. All right, so here is a map of all of the sites that currently have series with 9 or more years of data across the National Phenology Network database. And the sites with animal observations are in red, and those with plant observations are in blue. And you can see that we have over 3,000 individual series of more than 9 years, which is amazing, on almost 450 different species and over 400 different sites. Now, if you exclude any observations that do not have a prior no observation in the last 7 days, so the most temporally precise observations, it gives us many fewer series, but it's still over 900 individual series, 252 species, and 183 sites, which is pretty impressive. And you can also look at the how these differ by life form, right? So trees, shrubs, herbs are the most commonly recorded types of species in these data sets, but there's also a number of long-term observations of bird species, insects, herpetofauna, which is just reptiles and amphibians, and even mammals. All right, so what do we see overall? Has the phenology changed over the recent past at these monitored sites with 9 or more years of data? And overall, across all species and sites, we're finding that spring phenological transitions have advanced about 1 day per year over the past 10 to 15 years. And this graph shows how it breaks out by life form. So these dots show the average phenological change over time for each life form, and the lines extending out from the dots show that credible interval or variability around that estimate. And you can see that the estimates vary by life form group. Overall, it is an advance in phenology. And these trends are actually fairly similar, even when observations with lower temporal resolution were excluded from analyses. So here are only observations with a prior no in the last 14 days, or observations with a prior no in the last 7 days. The overall trends are somewhat similar. You see greater advances when you start excluding more and more observations, and you see that variability increases as you reduce the number of observations to get those averages. All right, so phenology advancing about a day per year, and you can even see this in this relatively short span of time. That's interesting. And now we want to know, has the climate changed over the recent past at these monitored sites? So again, a shorter amount of time to look for climate change in most studies. But here's a map of all of those sites zoomed into the contiguous US, and each site is colored by historical temperature with warmer colors indicating warmer sites. And we do actually see a slight but significant increase in annual temperatures across all these monitored sites over time. And this graph shows that slight increase in mean annual temperature. Each line represents a trend at each site. And we're actually also seeing temperatures increasing at a significantly greater rate at sites at higher latitudes with cooler climates compared to sites in warmer, lower latitude regions. So this matches some of the results that we just saw from Dr. Gralnick, and it also matches results from global studies over longer timescales. However, we don't see any significant trends in yearly or seasonal precipitation across these sites over the past 15 years. These points indicate precipitation at any given site. It's very variable, but there's no trends over time. All right, so we've got phenology advancing about a day per year, temperatures increasing a little bit over time. Now we can look in depth also, especially with plants, because we have the most observations for them, at how different plant phenophases might be changing over time. And here we're just going to look at that finest resolution data, the 7-day interval observations. And for plants, over time, we're seeing all plant phenophases are advancing, although not all of them significantly, with leaf bud burst advancing the most significantly. And leaf senescence advancing a bit too, but not quite as much over time. We can also look at how they change with different spring temperatures. So in warmer springs, all plant phenophase observations have advanced more than over time. Again, leaf bud burst is advancing more and more precisely than the other phenophases in leaf senescence. And these sort of differences between the sensitivity of the phenophase transitions over time and with spring temperatures got us wondering if these differences could influence the length of the growing season, and if it was possible to observe that using data from Nature's Notebook. So to try and do this, we looked at site level data where there was an average leaf bud burst and leaf senescence date for the exact same species and site with 8 or more years of data to try and include a little bit more observations here. And this is a map of all of the sites where there are tree species with both leaf bud burst and leaf senescence dates across the Nature's Notebook. And we first looked at if that was changing over time. So the difference between leaf bud burst date and leaf senescence date, let's just call that leaf season. And there was a very slight non-significant increase in leaf season length over time across all of these sites. But there was a very strong 3-day significant increase in leaf season during warmer springs, which makes sense, right? It seems like leaf bud burst gets much earlier during warmer springs, lengthening this leaf season. And then interestingly, we find that the tree species at historically colder sites at higher latitudes extend their leaf seasons longer with warmer temperatures than trees from warmer, more southern sites. So this is getting again at that difference that we're seeing in phenological change across latitudes, which is really interesting and is only possible because you all are taking these observations on both leaf bud burst and leaf senescence dates for the same species over time at the same sites, which is really exciting and important. And I've got like a bazillion other things I could say, but I'm just going to end here since we're almost up against that hour. And I realize I've already shown a lot of graphs. So anyway, your observations are important, and these results, although preliminary, are really exciting.
Thank you so much, Janet. Yes, when we started to look at your results over the last few months, we were so excited about those patterns that were emerging that even though the data haven't been published yet, we really wanted to get everyone, give everyone a first look at those and just show the value of those really long-term data collection sites. So I hope everyone who's on the call, if you're a Nature's Notebook observer, you're comforted by the fact that there are really interesting patterns that could emerge from data, and hopefully that gives you some incentive to keep going if you're just starting to do observations now. So just maybe in the last minute or two we have here, I'd love it if any of you would like to share any advice that you have for observers based on your experience in looking at the data, or just anything you want to share about anything that was inspiring or anything that was challenging about working with the data. Whoever wants to go first.
I guess I could go quickly as long as I'm back on. I got shut it off of this, but I'm back in, right?
Yeah, we can see you.
Okay, good. I think one of the issues is it's difficult to focus because there is so much data on Nature's Notebook. And I think as a plant ecologist, I often am drawn to the plant data. And yes, there is more plant data than for other species. I was really surprised and impressed at how many other animal and insect species people are recording data on. And if you do really want to get at that idea of phenological mismatch or changes in these trophic cascades over time, collecting data at all these different levels and life forms is really important.
Oh, that's a great plug for adding an animal or two to your site if you're just doing plants.
I think Janet covered like a really important part of it. Like, you know, we one of the challenges with like phenological mismatches is it's really hard to get like direct information about like visitation of an insect to a plant, and that interaction data can be hard to like derive easily from like really any source. And so the more we have those paired observations at a spot at a time, the more power we have to like know what's happening. And so I'll just also say like repeat sampling, like it's just super important and valuable. And like you guys who are out there looking at intensity and giving really, really detailed reporting, yeah, we can get some data off of iNaturalist about whether there's a flower or a leaf or a breaking leaf bud on a tree or on a shrub, but like the kind of data you guys produce is incredibly rich, and that enables an enormous amount of science to happen that can't happen in any other way. And it's not going to happen from remote sensing, you guys, because I love the remote sensing record and it's really important, but you don't get like an observation of an individual. You get a pixel telling you it's like green from the top of the canopy, right? So what you're doing out there is making a huge difference in our understanding of like these big patterns, as both Janet and Ben mentioned. It's so I'm so thrilled and so amazed at what you guys do.
Thanks, Rob. Any last word from you, Ben?
I would just say to sort of build off Rob's point, the repeat sampling not only within a year, so being able to track an individual as it develops, as it moves through different phenophases, that's incredibly valuable. But just as valuable is the repeat sampling year to year as well. So sampling that same individual and looking at how the behavior, how the phenology changes year to year, or population, or whatever level you're making these observations at, that is also something that is fairly unique to NPN. We can't get that information from iNaturalist. Most people are just opportunistically sampling on iNaturalist. It's not the same individual or same population, and it's not something that we would even necessarily be able to tell if it's the same individual or same population, even if it were. So having that sort of that record really makes NPN data usable in a way that other resources, even other very similar community science resources, sort of lack. And it brings that data much more closer to the type of data that I collect in the field with my students. And so it comes a lot closer to the sort of professional quality data that we collect ourselves. And in some cases, it matches or even surpasses it. So it's a really incredible resource. And just to echo Rob, thank you so much for making these observations. They make my job a lot easier, and make the type of inferences that we can pull out of this data a lot better, and just a lot more interesting.
Thank you all so much for giving such great presentations. Hopefully you saw the kudos in the chat. People really enjoyed hearing about your research, and I think they're very comforted by all the examples of how their data are being used. So thank you so much for being with us today, and thank you to all of you who are attendees on this webinar and who have joined us all week long. We are hoping to do some more of these kind of webinars in the future. So thank you, and happy Phenology Week. Happy spring, if it happens to be spring where you are, and we hope to see you soon. Thanks all. Bye.